So, yes, I am going to deliver my lecture. After that, Professor then comes and he will deliver his lecture. So, today's topic is the metabolic databases and the utilization. So, yesterday I explained a few pre-processing steps of the NMR in which we use a few databases. Now, today you try to understand what are the different databases, which are the important for the analysis purpose, and how we utilize them for this particular task. So this information is just like a what is the latest information that is going on in the market. In the research area, you find out that ion mobility mass spectroscopy and data independent and the dependent acquisition drive are the innovative things which happened in the last 4 years. So you will understand very soon this point actually. So mass spectroscopy is told, otherwise I will come with another lecture after 2 of 1 hour and 1 hour of the data. So you try to understand this is the latest development that happened in this metabolomics, that is the ion mobility mass spectroscopy. And there is another innovative approach that come up very soon in the last 5 years, that is the data dependent as well as the data independent acquisition drive. So these are the two things which come very soon and they improve a lot in the metabolomics analysis. Yes, now questions come up that what is the application of the mass spectroscopy that we can use for the metabolomics. So the first thing that we are discussing here, quantification and the identification of the different natural metabolites which are present in the plants. That is the first application. Second, just latest you will find out. So we are just discussing what is the metabolomics. But you know, in the USA, they can find out the disease by just only imaging the patients. So they are working on the different patterns of the cell actually, and they are using some in situ hybridization process with the metabolites also. So by imaging, they can identify the many diseases. So that is another application of the mass spectroscopy. And yes, there is an issue. Within the cellular level, you can identify by the imaging, but you can also identify subcellular localization of the different metabolites. So we told overall metabolites, but now I am telling you on the basis of the imaging techniques, you can identify a specific pattern of the different metabolites which reflect the disease state of that particular cell. Similar subcellular localization of the different metabolites. Which metabolites is produced by which biosynthetic pathway and how it is utilized for the different biological process. So that thing can also carried out. And yes, the integrated approach of the metabolomics with the proteomics and the genomics and transcriptomics can give us the overall information of any biological process. So these are the few applications of the MS, and I am coming to the second point, that is the this is the our flow of workflow of the plant metabolomics. So here I will started with the sample. So sample you have to prepare. It is itself is a science, sample preparation. After sample preparation, you have to generate the data. And the data can be come from the GC and the LC. The technology which you want to use for your work, you have to use on the basis of the availability of the machine also. Some metabolites you can identify by the GC, some metabolites you can only identify by the LC. And you will identify, here I mentioned one PPT in which you find out we are using both the GC and the LC for the integrated identification of the metabolites, integrated as an integrated approach also. Now the second question is that we have to convert the file into more friendly file. So every machine is giving very complicated data and comprehensive data of thousands of the metabolites. So we can't handle them, and we can't have some machine which can give us so many analyses. So we have to convert that file into the different extension. So MZML is a most common, MZXML is also a very most common file which is used in the metabolomics. So after that we have to clean the data. So for the cleaning we have to use, what is the meaning of the cleaning here? Base corrections, signal distractions and necessity intensity filters. These are the few parameters which we can improve by using the cleaning of the data. The another one is that we have to identify the features which are associated with that particular data. So that we can identify based upon what type of the biology we are investigating by using this particular experiment. So now, as in the last slides, last presentation, I told you that there are the two different peaks. If you doing the improving something, so these peaks can come very close to each other. One is here, one is here. But when you are aligning the spectra, so both the spectra, these peaks come very close to each other. So alignment of the sample of the different set of the experiment is also necessary. In the one area, you find the peak somewhere, and another area, another graph, you have the peak area somewhere. And according to they are the different. But if you have the list of the metabolites, which is reflecting that this is that compound, then alignment is already done. After that, you have to go ahead with the validations of each and every whatever you are getting. So this is the most laborious thing actually. You have to validate each and every metabolite. So that with the compound databases. So you have to cross check it. It requires the time. Each and every metabolites you have to validate. Otherwise, how could you know what is that compound? And after that, we have to identify the different chemical compounds in the targeted and the non-targeted. Then we use the statistical analysis, and then you come on some biological interpretation. So this is the overall flow of the plant metabolomics that is reflecting here. So now, I'm coming on the another points. So, यहाँ से मुझे बिल्कुल नहीं दिखाई देता। So I am coming now. This is the overall picture of the metabolomics, but if you identify now, there are new innovations which come in this particular area. The first innovation is that this pharmacometabolomics is basically used for the identify the lipid droplets for the cancer cells also. So cancer cells have some specific type of the metabolism. They have some specific lipid droplet to provide the energy. So Raman guided actually, pharmacometabolomics is utilized for this particular purpose. The second is that, yes, every compound, so many phosphates are used for the different purposes, starting for the DNA replication, ATP consumption and many things. So now we are able to use P31 NMR. Previously we used the proton NMR. So now it is come in the fashion. You can go ahead with the P31P NMR for a metabolites which has the phosphate group in there. So now you don't need to go ahead and work with the proton NMR also. You can go ahead with the P31 NMR. That gives the resonance of the phosphorus molecule which is present in the different metabolites. Then there is another thing, that is the isotopic fine structures for the different molecular assignments is also just recently come up. So now it seems very easy for us to do metabolomics and analyze interpret the data actually. So automation is also come up, many reference materials, harmonization of the samples can be carried out, you have to compare your data with the others. And now there are the two different phasic method actually, monophagic and the biphagic. So basically we are shifting towards the monophagic methods for the faster workflow and the automation here. So I am coming the another points, that is the data analysis, automation and the resources. So here we are using the two different approaches. First is the machine learning and second is the deep learning. So we are using both of them actually, and this artificial intelligence is also come up in the picture right now. So by using machine learning and the deep learning, we are able to predict the things also. So you know, they improve the annotation, metabolites networking can also be carried out. So I will come on the example of the loinness. I will also provide the scripts of the loinness, also enhance ranking models for the small molecules. So what happening actually, sometimes we have to rank the different metabolites. So if we improve the ranking model, so which are metabolites which are present in the small amounts, they will also assign some rank. If any metabolites is getting a rank, so it is possible for us to use those metabolites for the different interpretations. Yes, new softwares come up, and omics tool is one of them, Crane, bioconductors, PyP, GitHubs. These are the different software actually. Most important is the FAN laboratory databases. FAN laboratory databases is working on the plant metabolomics, and FAN is the father of the plant metabolomics. So that was not mentioned in this slide, but yes, other than GitHub Labs is also has published many good software also. And yes, you have to, there are so many tools for the QC, quality control, then annotation for the visualization and the data formation also, databases also. So these type of the things is helping us to get the good coverage, better understanding of the metabolomics. So I am just coming with one example in front of you. Here you can find out what is the objective of this. Here the objective is to identify the key metabolites in the pathway which are involved in the maize drought response. Suppose this is your title, okay, you are working, and this is just like an objective of your PhD. You know? This is the objective, so you just use the similar type of the objective for your PhD synopsis. So here what we have to do? We have to use the monophagic and the biphagic extraction method, but here we are going with the monophagic extraction for better coverage of the metabolites. The second point is that what we have to do? Okay, we can use the Raman guided MS that detects. Raman guided MS can detect the imaging of the small molecules. So we are focusing here on the lipid droplets. So these lipid droplets can give us that some part of the cell is stressed and their occurrence and their non-occurrence is directly related to the response of the cell with that particular stress condition and healthy conditions. The another points that we can use is the P31 NMR metabolomics to identify the phosphorus containing metabolites, and yes, they ensure the precise metabolic annotations also. So analysis purpose के लिए we use the machine learning models. There are so many models we can be constructed databases for the dot sample and the highlight the biomarkers. Loiness is a network that it is a software itself. Loiness markers. So the proline and the lipid metabolism is highly active during the stress adaptation. So it means that if we have this objective, so finally we have to give the final result in the form of this particular thing. So these 4 different machines, for 2 different techniques and 2 different outcomes of this objective, we can go ahead. So it was the problem, and finally we have to find out that proline and the lipid metabolism pathway is highly active during the stress adaptation. So this thing we can do. The 4th point that come up about the data visualizations. So you know those compounds are upregulated and downregulated. And she asked, now what she do? So now you have to go ahead with the data visualization. You have to represent your data in the form of some real change actually. So if you go here, so I would like to recommend here the tool that is the most utilized databases, that is the Metabolomics. So these Metabolomics definitely give you some key patterns during the heat map, PCA and pathway maps. So if you know that this is your metabolites, you are detected in your particular method. So now the next step is the visualization. In the visualization, you can use anything which can create some classification, which can create some cluster, which can help to interpret the biological information. So here, heat map, you have to construct PCA plots, you have to plot along with the loading patterns, and you have to go ahead with the pathway maps also. Finally result is here that proline accumulation, lipid redistribution, and ATP reduction play critical role in the drought adaptation and future development of the drought resistance maize. So this is just like a final outcome of this one. So these 2 slides are giving the better understanding how we use such type of the things for our work. This is the loiness. This is the you can use it for your work also, and this is the metabolomic. This is something that can help in the visualization. So someone if ask me where from I can do the heat map, from where I can use the PCA, from where I can analyze the data. So you go and find out metabolomics is useful for you or not. This will be very useful for all of you. So I am coming on the other points. What are the different platforms for the metabolomics? So now I am shifting myself in the different metabolomics. So metabolomics has many techniques, and we are using the many techniques. That is the LCMS, that is the GCMS, that is the capillary electrophoresis, proton NMR, C13 NMR, P31 NMR, Raman FTIR, So every tool, these platforms are releasing the data in the different format actually. Now you have to use some tools which are highly specific with that particular techniques. Then only you are able to analyze the data. So here you can find out, we explain the NMR based tool only. Here that is the ALPS NMR, Sigma NMR and the NMR filter. So I am just giving the name of these two ones. I forget actually. So that's why I come with this thing. What is the name of this NMR filter and this thing? So give me. So ALPS NMR is the automated spectrum processing system for the NMR. This is the automated spectrum processing. Whatever the spectra you are getting, they automatically process it. So now it is easy for you. You directly put your NMR here. The second point here, we are using R package. So R package is a script actually. So it has step 1, 2. You can't move from the 5th step directly. There is a difference from the one step 1, 2, 3, 4, 5, 6. Okay. So this way, so that's why this R package has 5 to 6 different scripts starting from the one and finally you can use for this NMR analysis. So NMR signal processing can be carried out including the alignment that I told you. Second is the normalization, meta handling for the untargeted metabolomics. So this software ALPS NMR is enough for the pre-processing of the data. Second data, that is the Sigma data, is the signature mapping actually. So this Sigma data is the signature mapping. So it converting the raw proton NMR urine spectra. This is highly specific with the urine actually because this work is highly developed in the human medicine actually. So in the plants still we need so much because plants has so much diversity. This specific disease cell has a specific type of the spectra. So it is easy to visualize those spectra. So this Sigma signatory mapping you are mapping some particular catalytes, metabolites by using some particular tagging. So here we are using that signatory just like a urine actually. Urine spectra into the metabolite tables using automatic peak picking and a chemical shifting library. So that can be used for the NMR processing also. Third one is the NMR filter. NMR filter means they have this interactive software which has high confidence NMR compounds identification using the chemical shifting and matching algorithm. So everything is present there. NMR filter. You use the NMR filter, they can give the very good variables which can be easily visualized, identified actually. So these are the 3 tools which is basically used for the NMR. Now I am coming to the other points. That is the GCMS based tool. So GCMS based tools are little bit different from the NMR tool because they are associated with the GCMS. So GCMS tools में एक सेकंड में नाम मैंने यहाँ पे लिखे कम है नाम तो इसलिए मैं आपको लिख रहा हूँ. GCMS tools, we are differentiate all the GCMS tools basically in the 2 different categories. The first one is the MS Hub, EIGMPS molecular networking. So this platform has enough ability to store, processing and molecular network of the GC-HRMS data, high resolution mass spectroscopy. This is not only the GC, it is MS also. So both are the integrated. GNPS massive for untargeted MS data repository. The another one is the GNPS massive. So this is the public repository of the untargeted mass spectra and EI spectra data. So this is the public one. It is present in the public, it is available. So whoever is working of some untargeted metabolites, they must have to check those type of the databases because you will identify some validation from this particular place also. The another point, that is the 2D GCMS tool also. So 2D GCMS tool is also used for the GCMS. It is the pre-processing of the 2D MS data and basics are the same, that is the baseline corrections, smoothing, peak alignments, and yes, advancement of the data is reduced and we can further use it for the downstream analysis purposes. So this is all about the GC based tools. I am taking an example. so that you can easily understand this concept. So this is an objective again come up in front of us, that is the understanding tomato metabolomics, response to the pest attacks by identifying key metabolites involved in the plant defense. So now you have to identify the metabolites which come due to some pathogen attacks. Okay, now we will use both the two, NMR and the GC here. So the first approach, we have to use the NMR. That is possible for you. NMR and GC, it's easy to identify somewhere from where you can get them. So NMR, we use the ALPS NMR that I told you for the untargeted NMR spectra, processing to detect the stress-related metabolites like the alkaloids and the flavonoids. Okay, ALPS से आपको पता चल जाएगा. Then you apply the signatory mapping to map the NMR signal to a database, already published database, to identify of the stress markers. So you can identify the stress marker also. The second, we have to move from this NMR analysis to the GCMS analysis. So GCMS analysis, we have to go ahead with the, utilize the MS Hub EIGPS to analyze the volatile components linked to the pest response, creating the molecular network for the compound annotations, and use the RGC plus GC toolbox for the 2D GCMS to pinpoint, track down a particular defense-related volatile organ component. So with very high good resolution basically. So this slide is itself explaining that how good objective you have to design for your PhD related to your metabolomics, and which machine or which tools you have to use for your work. So now, just GC and NMR is giving the different type of the coverage. This NMR is giving the coverage of the secondary metabolite that has just alkaloids and the flavonoids, but this GCMS is giving about the volatile organ components. So now, after doing this work, you are covering both the flavonoids, high molecular weight components, as well as the low molecular weight components also. So now I am coming to the third part of this objective, that is the data interpretations. So when you use these tools, so you collect the data, now you have to integrate the data which is coming from the GCMS and the LCMS. So when you integrate the data, so you have to correlate the specific metabolites such as the alkaloids and the terpenoids and whatever the cyclophenolic compounds you can say with respect to the pest-induced stress. So along with this, you have to go with the control also. So control is a little bit different. Here you will identify some metabolites are upregulated, some metabolites are detected, some are not detected when there is a pest infection, or something like that. So now we have to do network analysis. So network analysis I'm not using here, but I just have to know what is the network analysis. So network analysis is just like a Facebook friends actually. So when you are making a call, so you will identify he is also present on the Facebook, and do you want to make the friend him or not? Okay. So if there is some events happen, so it's easy to identify, track down their friend circle. Okay, acha, he is the friend of him, he is friend of her, this way. We can identify, track down those things. So basically these things are linked with each other. When we have to explain the things with respect to the two things, me and he are the friend, so I am just like a node, he is also a node. Now, what type of the relationship we have, that is labeled as a edge. So everyone has a edge. So yes, we can, these data interpretations, networking is basically just like a graph actually. But it gives a better explanation of some relationship between the different objects. Me and all of you are the objects. Okay, how we link with each other? Maybe class, student, teacher relationship, friendship relationship. So we are the objective. How? On what basis we are linked? So that is labeled as a edge. So we can draw the network actually, and you can draw the network in the Facebook from the last. Who are your top 50 friends? So this Facebook will draw on the basis of some information like, dislike, comments, or something like that. So they create a network in which that network is explaining some particular information. So here also during the data interpretation, we have to create the network by cytoscope and some other software to highlight the role of the K metabolites in a particular pathway which could be used for that particular analysis. Here we are explaining that how network analysis helping that jasmonic acid pathway is behaving as a defense during that particular situation. The final outcomes, so what type of, everyone will ask that what we have to show and what are the things we have to provide as a display in the final figures. Because it is very important for all of you in your PhD and your MSc, what the data you are presenting. So for that I have a humble request to all of you. First of all, you read some old paper and you identify what, please in the paper, read table of every day. If you are reading only text and you are just skipping the tables and those figures, so you are losing more than 75% of the information of that manuscript. So this is my humble request. When you read any paper, scientific paper, so give more attention for figures and the table at least. So then you can analyze, okay, how they draw that structures, what are the parameters, what type of the visualization techniques they use. If you try to focus a little bit on those type of the things, so you know what type of the visualization you need for your work. So that visualization is necessary for here. Final outcome of this objective is that we finally identified that jasmonic acid derivatives and other volatile organic components are the primary K metabolites for the defense purposes, and they provide the defense against the pest resistance crop breeding. So this is the same thing, data spectra and the metadata interpretation. We have to exclude some regions, we have to focus on some specific area, then we have to find out what is the outlier actually. So some outlier come automatically, we have to avoid those outliers, otherwise you will go in the wrong direction. And finally, peak detections, peak alignment you have to do, normalization of the data is another necessary steps, and peak integration, and finally you are able to data for the machine learning actually. So machine learning you have to do, and this is the before that one. So these are the 4 steps which are necessary for you. It was the objectives, and finally you are able to go ahead with the machine learning. Machine learning is some machine has some specific instructions. Whatever the data you are giving, so they do automatically. Predictions, some neural methods, network is also present here. So you give, they will do. Even they can classify the metabolites also. So now I am coming the another tools which are necessary for us. Now I told you 2 different experiments and how we use this technology for these 2 different experiments. Now we are coming on the tools actually. What type of the tools are here and how they are useful for us. So the first tool is the pre-processing and the quality control, QC control and the pre-processing. Yesterday I also told about the NMR pre-processing, how the 5 and 6 points are responsible for the particular pre-processing actually. So yes, here you can identify, we have to start our work with some task. What is your task actually? Our final task is the feature extractions. We have the data, big data. Among them we have, otherwise everything is ABCD XYZ. You have to extract the feature from that particular data. Then second, whatever the feature you are collecting, you have to visualize those things in the, whatever the chromatogram. Chromatogram is just like a graph actually. It's a final outcome of the chromatography. So whatever the chromatography you are using, the final peaks that coming here, that is we are labeling as a chromatogram. So how your chromatogram is visualized? Is there any other RT values or anything? You have to optimize that. Then you have to come on the peak alignments. And then finally you know the deconvolutions, means extension change of that particular original data into some more user-friendly data, then page correction as well as the blank correction also you have to do, normalization of the data and the quality filters actually, QC, quality check. So these are the few steps, key tasks we have to carry it out, we can carry it out by using the pre-processing data set tools. So whatever the tool I will mention here, they are doing some task of this particular filter. Now what are the major challenges is that we need the computer which can handle such heavy big data. Second, what is the efficiency of our machine, and are we able to understand the outcome, input files, interface must be very user friendly, otherwise if interface is not user friendly, so we are taking note of the problem. And what is the advancement that is coming every day, it must be compatible with our data analysis. Now another point that comes here, every software has some positive points and some negative points, and they have some errors in the form of the systematic variation and some mismatching. So we have to understand those things, and we have to avoid such type of the software incorrect variations to analyze the data. Now I am mentioning the name of those pre-processing tools. There are so many. We come up with only just a few for your better understanding. So I will explain each and every one, and these are basically scripts. So I told you we have to have some particular task of our work. The tasks are representing here. The tasks are the selected task, that is the feature detection, program visualization, fit alignment, deconvolution, base correction, and the blank corrections also, normalization in the QC. So now all these can be carried out by these particular pre-processing tools. The first tool was the crop RPSS package. So this is the post-processing tool actually. It is used after processing even to remove the redacted LCMS features. So when you are extracting the feature now, so you will identify some features are come up, but they are not useful for you. They are making some wrong interpretation. So you have to remove those features. So this tool is helping to remove those type of the feature. That is the crop features. And yes, they group the different components, metabolites, and they can correlate the different features also and visualize the correlating network also. So for the correlation purpose and for removing the non-essential features, this package is very useful. The second is the NCGTW. NCGTW is the reference free alignments actually. So this is from the XCMS. XCMS is working from the last 10 years, 1 decade, and it is working very fine actually. So this NCGTW is basically used for the XCMS and is correcting the misaligned feature group in the LCMS data. Sometimes there are the 5 groups, okay, and we aligned the wrong feature with the wrong groups. So we have to remove those type of the things. So for that we can use this particular tool. The other is the TDMS. TDMS is a Python script, and yes, it is pre-processing the LCMS data with functionality of for normalization, imputation, QC correction, as well as the visualization also. And the another important point is the MetampX actually. MetampX is so many tools actually. It is not a single tool. It has 103 metabolomic tools, and they streamline all the untargeted MS workflow. So this is just like a very big flow. All the tools are synchronized, and you use them systematically to find out the untargeted metabolites. So this is the pre-processing tools. There are so many. We are just only focusing only a few for the sake of the easiness otherwise you will be boring for so many tools. The another most important point is the QC control actually. QC tools. You have to validate the data actually before to process for the another steps. So for that we are using the R package signing, that is the HRUV. HRUV is the remove the unwanted variation among the LCMS batches. So batch, there is so many batch now. You are one day you are running, next day you are running. So maybe there is some variation come up. So to remove those variation, unwanted variation, so you in between the different batches, we are using this particular tool actually. And remember, the LCMS batches can be easily visualized by the PCA, RLE, and the run flows. So when in the PCA you are not able to identify what is the issue, you identify that there is some metabolites which is not coming in that particular groups, and they are the part of that group. So maybe that due to the batch differences. So you go back and use this tool, remove the unwanted, and then you have to run again the PCA. You can remove those type of the unwanted peaks also. The another is the MetaQC. MetaQC is always Biocorrect's kits actually. This is the whole kits which can give the visualize the reports and summary status. So this is easy for us, but yes, these are the R package. R package means the script language which is used to construct this software is the R actually. So DB norm is another one, and yes, technically heterogeneity is come up. So what happening actually, sometimes we use some parameters and some due to that some technical errors come up, and due to those technical errors many times you are not able to capture those technical errors. So we remove the technical errors by using the DD norms actually. And the last one is the meta clean R package. It can assess the peak quality actually in the untargeted databases using 11 metrics actually, 11 different parameters by using the machine learning algorithms. So if you just open it and try to understand and read what are those databases. You know which one is the used one, best one. So you have to use one for the different objective, you have to use the different tools here. The another important point is that I am giving the workflow for the pre-processing and the QC in the maze metabolomic. So this is the workflow which you have to use. So the first objective is that identifying the drought responsive metabolites in the maze using the untargeted LCMS and the MS. So here we have to identify the drought metabolites basically. So raw data, you know, as I told you, .mz.ml is the raw data file actually. This is the extension of that file, and we have to use the first software, that is the TDMS. TDMS is convert the raw LCMS data to the Z file. Then we have to align the peak normalization and the visualization. So these are the things we are doing in the TDMS. So after TDMS, we have to go ahead with the crops. In the crops, you have to remove the redacted features with high correction in the RT windows and visualizing clusters also. The another, we are using the NGGT. Now you can understand, we are using those things in the pipeline because one is giving the another information, second is giving the another type of the information. So we are using the NCGTW. NCGTW remove all the redacted feature, whatever we observe during the process, and we correlate them in the RT retention time windows, and then we visualize the different cluster again. And then you can use the HRUV, then MetaClean, and final K marker come here. So everyone is used, but there is a pipeline where in which you have to use one database before to another one. So HNRU gives the better correlation between the inter and the intra-batches variation. It can be validated by the PC basically. And MetaClean can be do the machine learning, filtering of the poor, ignored, integrated peaks, and we retain the high quality peaks. यहाँ पे poor को निकाल देंगे, high बच जाएगी. So finally you can identify the K biomarkers, which is our objective basically. So here your job is done here if you use this one. So we explain the things in that way so that at least you are able to understand how we use those type of the databases for our integrated approaches. Now what are the challenges for these type of the things is that yes, challenges has integration of the orthogonal data is a major issue with this type of the problem because you have the limited RT predictions with the MSMS for improve the annotation accuracy. So this is a major problem because you have to combine the retention time prediction with the annotations actually. The another point is the database coverage. So current database cover only the 5 to 30% of the detected feature in the untargeted databases. So untargeted databases you have to, what the feature you have to use actually. So its coverage is very low, even till today. Whatever the databases you are using, they are not giving the full coverage of the untargeted database. Only 5 to 30%. So try to understand what is the scope in the near future in this particular area. FDI management is another point actually. So you have to balance the high quality annotation with comprehensive feature coverage also, and standard developments. Yes, many community are working on the standards to improve the reporting consistency. So they are spreading their databases to the different community. They are getting the feedback and they are improving. So this is the continuous process that is going on actually. So now I am coming on the another point, that is the annotation tools. So because we know the data, now you have to identify the things, now you have to allocate. So for that basically ion mobility, DIE and CCS are the new technology which come just only 4 to 5 years before, and they can improve the annotation, but there is another challenge come up, they induce the complexity. So you have to again add the complexity by using this thing. So the same thing that I told you, because the database is 5 to 30%, the same thing come up here, that is the limited annotation, and this limited annotation is due to the gap in the databases. So now it is very difficult. And lack of the integration between, this is the same challenge that we saw in the last slides. There is no better integration between the retention time predictions as well as the MSMS data. It still remain a big challenge for all of us. So what is the proposed thing that we saw here? So these are the metabolic annotation tools, substructure annotations, visualization and the cleanup, then pathway and the biotransformation, and the fourth is the compound identification, fifth is the GNPS tool, and the retention time prediction. So here you can go ahead with the many tools. I'm explaining the name of those one lines actually. I forget. And this paper and the slides we will give you. We will give you such slides, no issue. हमने भी तो कहीं से बनाई है ना. Yes, sir. Give me 1 minute. I'm just smart, metafile, annotation. Yes, this full name of the MESAR is the Metabolite Substructure Auto Recommender actually. So this one is a web-based tool actually. So many, now these two are the web-based tool actually. An automated method for the substrate recommendations guided by the association mining, and this suggests the substructure from the spectrum library. So what is the library generating? It can identify the substructure, and this MET-FID is another one which is generally using the neural network for the molecular fingerprinting. So these are the two things which is basically used for the substructure annotations. The another point is the visualization and the cleanup. So visualization and the cleanup we are using the CPVA and we are using the MS-Clean-R. CPVA allocates the LC peaks and reduce the noise basically. And the another one is the MS-Clean-R that filter and allocates the LS data, MS data, removing the redundancy. So visualization is necessary. So before visualization, you have to clean up the raw data for the better understanding. The pathway and the biotransformation is another area where you have to, pathway enrichment analysis you have to carry out. You ask now what type of the figure you can make. So every one window is reflecting one and the two figures. So if you are working on a metabolomics or a particular issue, you can create, if you have some biology, you can create easily 40 to 50 figures. So substrate annotation can give you the two figures, visualization and the cleanup again give you the two figures. If you are adding some biology, so you will get many other figures. Pathway and the transformation, that we are using the MET-ESP and MET-IDDFR. So you can predict the drugs metabolism also by using the MET-IDFYR actually. So compound identification is another area for the annotations that we are using the Canopus. Canopus can predict the compound class with the deep learning actually. So here we are using the deep learning concept and we try to identify it. The class at least from which those metabolites are belong. And more discovery is very efficient database search actually. Yes, you have to search many databases. So we have to use some program which can give us all the information about the different available databases. So more search, more discovery is useful in that concept. GNPS tool is the chemistry that is link MS data with the chemical ontology actually. And IIMN is giving the connect related ion in the molecular network. And retention time prediction it's a machine learning program and it can predict the retention time of a particular metabolites for the LCMS databases. So these 6 different windows are representing the different annotation tools, and every window is representing a particular figure of your work also. So that's why these are the annotation tools. So this is just like an illustration here. Here illustration is showing that how you go ahead and obtain the seed metabolites. So seeds are easily available and you can start your work with the seed metabolites. In the seed metabolites, just try to understand I'm not able to see here, so I will speak from here. So MS table is the first thing you got from your LCMS. Then you have to MS file. It comes with any extension and that value must to charge ratio. Now you have to pass by some specific conductors. That this conductor where you have the extraction features. So MS1, then RT, MS, these are the few things. Finally, you examine a few metabolites which are present in the seeds. So this is the first steps you are carrying as a step 1 actually. So now step 1 has the two different phase, but either it will go in the step B or in the step C actually. So when it is going in the step B, so we have to do the global metabolite annotation. The first A is the seed metabolite annotation. Now we have to find out global metabolite annotation. So you know what are the unknown components, what are the known components. You have to differentiate your whole sample into the two different categories, and these two different categories you have to work with the confidence level now. So confidence level 1, level 2, level 3, 3.2. So this way you have to classify the different metabolites. Okay. How much confidence you have and how they are useful for you. The same thing we have to come this direction, seed metabolites. So seed metabolites is coming by this way. So what known as guided multi-layer network actually. So we have to do this thing again and again. First of all, what are the knowledge of those metabolic networks? You have to search from the literature, what are the known, what are the unknown, what are already established. Okay, so suppose they are the 5 to 6 compound and you know the different reaction by which they are catalyzed, they are produced. So you do this thing and annotation unknown compound from the seed metabolites by using this thing. Here that saffron color dot has so many blue and the red color. So red color triangle are the unknown while these seeds are the major components and this unknown are the known are the blue color. So now you have to identify what are the unknown metabolites. Are they already published or they are really novel actually? So you have to do the reaction pair, you have to do the MS similarity concept. Now on the basis of that you can find out that a few are associated with each other. So here you can find out that yes, there is some challenges also come up that what is the predicted retention time based upon the machine learning, what is the surrogate mass. Surrogates means you add some compound before the extraction and identify what is their MS spectra and what is the retention periods. So on the basis of that you manipulate the raw data and finally you allocate the different known and the unknown metabolites with particular stress conditions or global peak. So here these are the abiotic peak and finally you identify that these triangle are associated with some other green color metabolites. So these are the abiotic MS1 peaks and there you can correlate the correlation and finally we go back and finally we produce this particular level 2. Here you can find out that what are the known and the unknown, they are linked with some other components. So this is a overall outline, multi-layered network KGMN which we can use for the identification of the known, unknown components by using series of the manipulations. So yes, I am giving here snapshots of these two and three different components tool which we are using, that is the NR Pro. Output on the demo data on the NR Pro, you can use here 5 compounds, monoisotopic mass, their formula, no ring, and these are the PubChem, what is the entry of these metabolites in the different, what is the score peak, what is the p-value. So you by this you can identify also that the redo analysis carried for the demo data you can do here, and this metabolomics workbench is also available here, that is the meta in web analysis result. So you can use these type of the software which are online available for your particular purposes. I am coming on the another snapshots of the subset of the tool, and the smart tool is basically for a chemometabolites MESAR for the corosolic acid and devo analysis on the FOBI. So these are the different tools you can use for your purposes. And now, what is the multifunctional tools for the metabolomics data analysis? So these multifunctional tools enable user to handle end-to-end workflow for the mass spectroscopy. So when you are using multiple tools simultaneously, so end-to-end data is going to analyze and finally you are getting the final output. So this streamlined process basically starting from the raw data processing up to the interpretations, providing a unified framework for the quality control, statistical analysis, data visualization and the annotation. So these are the multiple tools for the metabolomics. You can use anyone also. Skyline is very famous. It is expanded for the small molecules analysis with feature like peak picking, alignment and the quantifications. The second is the NOTAMI. NOTAMI is an R package for LCMS data for bit correction, QC clustering and the visualization. Balsam is a vacuum for the MCC, IMS and the LCMS breath analysis with peak detection as well as the alignment. MRM kit is automated peak detections. Metabolosiny is basically dimensional net reduction and the advanced visualization. Smart peak is basically ensure the reproducibility, and MS dial is basically specialized lipidomics tool basically. That is for the RT, CCS and the MSMS data, quantifying around 8,000 lipids actually. So recently I reviewed a paper of MS dial 4 actually. IP4M is a comprehensive untargeted metabolomics, and finally DROP-MS is a browser-based tools for the patromics and with the recalibration and the visualization of the different diagrams. So now I am coming with the snapshot of the IP4M for the GitHub laboratory actually. This is an integrated program platform for the mass spectroscopy based metabolomic data analysis. So you can just link and you can go into this particular multiple platforms. They are integrated with each other and you can use for your data analysis. What are the statistical analysis and the visualization tool for the metabolomics? So now I am coming with the visualization tools. So these tools offer a range of the functionality from the data normalization, integrations, advanced statistical analysis and the interactive visualization. If your visualization is not interactive, so how could the reader understands what is your finding basically? So they are designed to streamline the analysis of the complex metabolomic databases, improving data interpretation, enhancing reproducibility, and enabling deep learning of the metabolic network. So these are the different components. Yes, estimators, metabolite autoplotters, metabolite investigator, VIIME. So this VIIME can be used for the PCA plots actually. And lipid DR, then NOREMA, and RR Metabovas, and the JSMS2. So these are the different tools which can be used for the different purposes. But you can go ahead with the RR is basically used for the raw data analysis from the Thermo Fisher actually. And this JSMS2 is a cross-platform browser-based tool for the visualization and interacting with the MSI data in the 2D or even in the 3D also. So this is the web-based interface and the snapshot of the output for the demo for the VIIME, metabolomic autoplotters and the summer. So first one, you can do such type of the SC plots, heat map plots by using the VIIME plot software. The second here, you can sometimes metabolite autoplotters, you feed your values and you will find out such type of the graph actually, which is basically you identifying your figures. And the third one is that summer. So summer give the their size based upon their quantification actually. So some are small, some are the big. So you can find out such type of the visualization also by using the summer platform. So these are the databases for the plant metabolomics which are till today. Now I am coming with the plant metabolomics databases. You need them actually finally to validate your data. So profiling PCMD was the first one, which profiled 530 plant species with 2,13,264 metabolites from the 8,000 enzyme, and tool is basically used for the species comparison and the metabolite analysis. The another one is the PM Hub, comprehensive database on the plant metabolites, including the curative pathways, analytical method, and species metabolite profiling to add the research and the integration. The third one is the PlantSys. PlantSys is a part of the BioSys, MetaSys. Basically, this is part of the MetaSys actually. So it has 1,000 metabolites, 5,000 reaction, around approximately 5,000 compound, 4,000 enzymes, and extensively referred 15,000 citations. Try to understand the strength of the plant cyst basically. Methylene is the another one that is also very important for the plant databases. Actually, if you are working on the plant, so you must read those things, methylene. It provides the spectrum line of the LCMS and the GCMS data. If you are working on the LCMS and the GCMS, so you must go ahead with the methylene at least. And the RefMet is something that gives the data harmonization, particularly in the plant metabolomics, ensuring consistency among the data sets actually. So this is something. This is the workflow and the primary content of the plant metabolome hub actually. So you can go, this is the plant's material actually. So you first of all MSMS library, you have to find out the feature between the species here. You can find out the feature in the species. One, you have to identify the feature among the species, within the species, one in between the different species. And you have to do the reference network, metabolic profiling, multi-omics, and finally you have to prepare a network here. So this network by which you can identify a conjugate gene which is responsible for the whole metabolic alternation in that particular situation, biological situation. So this is the flow by which we can go ahead with the metabolomics, and this is the plant metabolomic pathway databases, that is the PCMPMN, and second is the plant comparatively metabolome databases. Because you have to compare among the different species also and within the species also. So for that you can go ahead with those two different components. This is the snapshot of the PM which is used in the plant cyst databases. Basically it is plant cyst, metacyst, biocyst, they are the same databases. One is biological related, second is related to the plants actually. So now, what are the specialized tools for the metabolomics analysis? So these are the specialized tools, means isotopic, if you are working on the isotopic work, so these are the few tools, Miami and the IsoScan. There are so many, we are just selected, we are representing here selected tools for sake of the easiness. For the lipids, if you are working on the lipidomics, so lipidomics is the lipidomics, lipidocreators and the lipid annotators, and a few more lipid profiling databases are already published, so you can use anyone for your work here. Now, mass spectroscopy imaging tools, so Raman to IMZ that I told you, it can convert the Raman imaging data into file IMZ, so you can use for the imaging analysis purposes also. Multi-omics integration tools, Samad and the Macropropagate is the another one. Macropropagate is the network-based approach, and emerging focus is basically, yes, still we are not able to find out only a few tools for the whole work actually. So we have to enlist all the different metabolomics, and we have to use the omics also, we have to find out some tools also. So another upgradation is a major issue actually. Every day you are getting a new update, so you have to take care. You have to update yourself also along with the upgradation. So that is another point. So this is the summary of our work here. So current tools basically focus on the annotations. So we can, whatever the tools are available, they are only covering 35% of the total annotation here. Data analysis, we can go analyze the data up to the 82%. Program language, yes, there are only 28 tools using the Python, 11 tools are using the Java, 5 tools are using the web-based, and the 3 tools. This is the programming language, the different databases which are used here. User friendly, around 50% they are the friendly, whatever the data going on. GitHub process is around 57% of the total tools are present in this GitHub. You just go GitHub and most 50% database can be curated. Active development, because every day you are getting the new updates, is a challenge, and specialized applications, you have some dedicated databases, you have to take care of those databases here. And yes, conclusion, that is the, you know very well, it's we need to get more precise accuracy quantification of the different metabolites. So, and this big data analysis is always a challenge, and when you are making some change, so it induces the complexity, we have to think about this particular way. And yes, these tools are helping us improving the utility of those data. We can identify the few biomarkers, And for the GNPS and this lipidomics is giving the better innovations. So in metabolomics, the best class in which metabolites quantification is going on, that is the lipidomics actually. So lipidomics has their dedicated software also, tools also, databases also. So they are present here, and this is the thank you all of you, and I would like to appreciate for your listening my lectures, and I would like to appreciate one of my students, that is the Sachit, who helped me a lot to prepare these slides. And yes, this presentation is complete. You can ask whatever the question, otherwise I am available here. You can ask me. This PowerPoints, everything you will get. But yes, it had, we explained here 3 different objectives and try to integrate the different pathways, the different tools in those objectives, so that you know how you get the final outcomes. So it was the special things that we introduced just one day before. You must know how we go ahead with such type of things. Otherwise, you never get the clear picture. You know these are the different instruments, but if you are not getting the clear picture how these different tools are used for the NMR, for the GC, and for other purpose, so I think there is no need to represent these things in this way. So we tried hard, and I hope it will help you give a broad view how you visualize the data, how you analyze the data, what are the different tools, databases which are available right now, and where these tools are present actually, and what is the script. So if you want to really pursue your career in the metabolomics, so it is my humble request to you, please go to any script actually. If you know the one script, and it's only just 20 to 30 days course of any script. If you know the script, so you can use those bioconductors and R languages for your work very well. And if you know the script, so when you are going and appearing in an interview, so you are really different from the others. So anyone can take you for their research, and if you know the script, so you easily design your own program also. And in the PhD, if you know the script, so definitely there is a chances that you can easily understand and utilize the different scripts which are related by the different persons. Second, there is more chances to get the postdoctoral if you know a single script language. So this is my humble request today, that you at least when you complete your PhD or your MSc, whoever is from MSc and the PhD, at least learn one script and try to use those scripts for your day-to-day problems in the metabolomics. So only after that you can do this thing very well. So yes, any question? I can. We have only 2 minutes, so I can take one question from anyone. Otherwise, I would like to hand over my. Yeah. Right.
तो कि ये सब टूल्स हम लैपटॉप कर सकते हैं, गेटिंग द रो डाटा, एनालाइज द सॉफ्टवेयर व्हिच आर इंस्टॉल विद द मशीन।
जो वेब बेस्ड टूल है उनको तो और जो स्क्रिप्ट है ये है कि आप है ये मशीन किस पे काम मशीन का लाइनेट वीडियो कैसे लगेगा। तो इसके लिए कि जिनका आप पता नहीं लैपटॉप कौन सा यूज करते हैं, तो विंडोज यदि आप इन चीज़ों पे काम करना चाहते हैं तो आप लैपटॉप लेके लाइनेक्स पे नहीं आता, मैं पहले बता दूँ। और यदि आपने काम करना तो आपको विंडो ऐसा लगेगा बड़ा बेकार। आप लेके आइए उस पर बस एक बटन दबाइए, लगेगा कि मैं इसको उठा के फेंक दूँ, एक बार उठा के लेके आइए। Try if you use the you can use anything. It's very cheap. Very virus no listen. Here every day we are getting a 3 data is cloned and everything is not safe in those windows. So when the liner can use it. Some scripts, some of the R language, and some of the other languages also script language. Many script languages are available. So you have to be prepared at least one script so that you can understand another one. If you know one script, so just take a few live lesson session online. You can understand the second script also. And if you know the script, you can develop your own program also. That is more useful. So I would like to invite the Professor James for his presentation.